Welcome to the deep dive. You've likely heard of gout, right? That awful arthritis feels like needles in your joints. It's ancient. But imagine if the fix for that, and maybe even other big modern health issues, was hiding way back in our evolution, just waiting for science to, well, bring it back. That's what we're digging into today. How scientists are using tools like CRISPR, the gene editing tech, to maybe reactivate a lost gene, not just for gout, but potentially things like fatty liver disease, high blood pressure, even heart disease. It's a wild link.
It really is. It's a fascinating journey into how our own biology, something that helped us survive way back when, is now paradoxically causing us problems. And understanding that history, that seems key to finding new treatments.
Okay, so let's ground this in the present for a second. Gout itself. It's intensely painful. It happens when uric acid levels get too high, forming these crystals. Not just joints, but kidneys too, right? Causes terrible inflammation.
Exactly. Uric acid, it's normally just waste. We have an enzyme, uricase, that's supposed to break it down. But if you get too much uric acid that's hyperuricemia, it's a red flag for more than just sore joints. Critically, high urate isn't only about gout. It's strongly tied to kidney disease, other serious issues too. Dr. Richard Johnson, who's done a lot of work here, he even compares the risks to like high cholesterol. And the numbers, they really tell a story. Something like 25 to 50% of people with high blood pressure also have high uric acid. But get this, for newly diagnosed hypertension, that overlap jumps way up, sometimes near 90%. It really shows how linked these things are. Suggests uric acid might be more than a symptom, maybe even a cause.
Wow, okay, 90%, that's a lot.
Yeah.
So this waste product connects painful joints to major heart problems. It really begs the question, why us? Why do humans have this vulnerability when most other mammals, they just don't? What did we lose?
Well, the key thing is, humans and other apes, we actually lost the functional gene for that uricase enzyme. Happened roughly 20 to 29 million years ago. Most other mammals, they still have it. It keeps their uric acid levels low. Our ancestors, though, went down a different evolutionary road.
And there's that really interesting theory for why we lost it, the thrifty gene hypothesis. Dr. Johnson and others suggest that maybe higher uric acid helped our early primate ancestors get more fat from fruit sugar, sort of like a survival hack, right? Storing energy efficiently when food was hard to come by. Brilliant back then, maybe, but maybe not so great now with, supermarkets everywhere.
Precisely. A huge advantage when food was scarce. And, importantly, this wasn't like flipping a switch. The gene gathered mutations over a long, long time. Its activity just faded. In fact, it happened independently in at least two different ape groups, the great apes and the lesser apes. That strongly suggests there was a real advantage to having higher urate levels back then, maybe for bigger brains, allowing for that easier conversion of fructose to fat.
Right, an ancient survival trait now causing modern problems. Eric Goucher calls it an evolutionary paradox, which feels spot on. And this is where the really cool science comes in, challenging that legacy. Professor Goucher and Leslie Mabalico at Georgia State University, their goal is pretty bold. Reactivate that broken ancient gene.
Yeah, their approach is clever. They used CRISPR-Cas9, people often call it molecular scissors, to slot a reconstructed ancestral uricase gene back into human liver cells. And liver cells are key because, well, in mammals that do have uricase, it's working hard on the liver cells, the hepatocytes. That's also ground zero for dealing with excess fructose and making fat.
And the initial results, they called them dramatic, which sounds promising. So in simple layers of human liver cells, uric acid levels plummeted. And crucially, it stopped those cells from turning extra fruit sugar into triglycerides, the fats linked to fatty liver and other issues. Plus, the revived enzyme even went to the right spot inside the cell, the peroxisomes. That's important, right? It shows it integrated properly.
Very important. It suggests it knows where to go and what to do.
And they didn't just use flat layers of cells, did they? They used 3D liver spheroids, too, like mini organs in the lab.
Exactly. To get a better sense of how it might work in a more complex, tissue-like setting. And even there, in those spheroids, the reactivated uricase still cut down uric acid and blocked that fructose-driven fat production. And again, it went to the right place, the peroxisomes. Now, it was a bit less efficient in the spheroids, probably because it's harder for things to move around in that 3D structure, but still, it worked.
Okay, so it works in these lab models.
Yes, and what's really neat is how these results circle back to that evolutionary idea. By reactivating uricase, they essentially reversed the very process converting fructose to fat that the original gene loss likely enabled. It strongly supports the hypothesis that losing uricase helped our ancestors make fat more easily from fruit, which was probably vital for things like brain development back then. It's a direct demonstration.
That's fascinating. So we've got this validation. What are the big picture implications then? Clearly, this isn't just about making gout less painful.
Oh, far beyond gout. Think about it. If you can safely lower uric acid this way, you could potentially hit multiple problems at once. Fatty liver disease, high blood pressure, cardiovascular disease. These are massive global health issues. We're talking about maybe tackling a root cause that connects a whole cluster of these chronic conditions, not just managing symptoms after the fact.
Which is huge, especially because current treatments aren't perfect, are they? Some people don't respond well to gout meds, or they have bad reactions to the artificial uricase therapies out there.
Exactly. There's a real need for better options.
Yeah.
And that's where this CRISPR approach looks so promising. By putting the working gene inside the patient's own liver cells, you might get around those immune reactions you sometimes see with external therapies. The idea is to get long-term, localized enzyme activity right where it's needed for metabolism, a more natural fix, potentially.
Makes sense. So the path forward, what comes next? Animal studies, I assume.
Yes, absolutely. Extensive animal studies are the crucial next step to check safety and effectiveness. If those go well, then eventually human trials could be on the horizon. And they're thinking about how to deliver it too. Could be direct injection, maybe taking out a patient's cells, editing them, putting them back, or even using those lipid nanoparticles, tiny fat bubbles basically, to carry the genetic instructions. Similar tech to some of the COVID-19 vaccines. Shows it's feasible.
Okay, sounds like a plan. But realistically, there must be hurdles. Big ones. Gaucher himself pointed out, didn't he, that genome editing still faces substantial safety concerns. It's powerful stuff. Needs to be rock-solid safe.
Absolutely. Safety is paramount. Off-target edits, long-term effects.
Yeah.
These all need rigorous investigation.
Yeah.
And then there's the ethical side. Gaucher also mentioned that once the safety is sorted, society needs to have some tough conversations, like who should and should not have access. If we can tweak our ancient genetic legacy, who decides when and for whom? It opens up a whole can of worms, really. Big societal questions, not just scientific ones.
Definitely. It's a deep question. This whole exploration really highlights that incredible link, doesn't it? How something that helped our ancestors survive millions of years ago is now a factor in some of our biggest modern health challenges. It really underscores how tied our health is to that deep evolutionary history. And like you said, it forces us to think about the ethics of changing that blueprint.
But it also shows the power of science, doesn't it? How researchers are using tools like CRISPR, tools we could have imagined not long ago, to potentially undo that ancient change. It offers real hope for treatments that could be genuinely transformative.
It really does. So for you, our listeners, something to think about. What other thrifty genes, these ancient survival traits, might still be lurking in our DNA, contributing to modern ailments we haven't even connected yet? And what future discoveries might reveal even more surprising links between our deep past and our health today? We'll leave you with that thought. Thanks for joining us on The Deep Dive.